Hi guys! So today we're going to be talking about DNA replication. So let's start off with summarizing what we did last video, which was discussing the structure of DNA. So typically DNA is in a double helix format, like this first part over here. And it has very tight bonds. We know covalent and hydrogen bonds between. So we have hydrogen bonds between the two nucleotides, and the two bases. Like base. Here we have hydrogen bonds. And between the nucleotides themselves, within the nucleotides, we have covalent bonds. So this is important to note when we take DNA replication into consideration, because we're going to... Oh, that rhymes. Consideration, replication. Okay, so because in the first step of DNA replication, we have to unwind these strands. So we have our first enzyme, which is topoisomerase. So if you haven't heard of this enzyme, that's okay. You might not have to know it. But the job of this enzyme is to stress the H bonds between the DNA strands. Because when helicase comes to unwind them, it'll make its job way easier. So that's the job of topoisomerase. And then after it does its job, helicase will come in and start to unravel. Yes, that's a chicken. And start to unravel these DNA strands to end up like this. Number one. Yeah. So once they're unraveled, it'll be easier to manage them and to replicate them, which is going to be over here in step number 2. Step number 2 is to replicate. Yeah, it's really that easy. 2 steps. So first, we're going to have our DNA polymerase. This is enzyme number 3. Enzymes are in green. Color coding, as you can see. So DNA polymerase is an enzyme, and there is a DNA polymerase 1, there's DNA polymerase 2, and there's DNA polymerase 3. So we're not gonna talk about those because I think those are HL material, but I'm not sure. But just in case, you should just know that they exist. Yeah, so that's what DNA polymerase is. It's a molecule that is going to help replicate the DNA strand. So DNA strands go in a 5' to 3' and a 3' to 5' direction. And for example, this one is 5' to 3'. This one, the black one, is going to be 3' to 5'. This just depends on the location of where, like over here, this is our DNA molecule. Our nucleotide, sorry. And here's our other nucleotide. Let's try to draw this backwards in one go. Oh, that didn't work very well. Okay, so this is going to be our other nucleotide. As you can see, it's backwards, the opposite direction, because it's flipped. And that's what gives it its dual anti-parallel structure. So that's how it looks. So as you can see, this is O. This is our first, second, third carbon. So that's why it's called the 3, 4, 5. So that's why it's called the 5' to 3'. And over here we can see it has this is O. So we count 1, 2, 3. That's the 3' to 5' direction. So it just depends on which direction the topmost C is facing. So we're going to start over here. Let me erase these. So DNA polymerase is going to work in a 5' to 3' direction. And that 5' being the 5' of the new strand, which will be the 3' of the old strand. So that's very important to keep in mind when building and replicating. So let's say this is our new strand. This new strand, so this is our old strand, which is called the leading strand. And this is our new strand, our beautiful pink new strand. So the DNA polymerase is going to come and bind over here. Let's make it a circle. So this is our DNA polymerase. It's going to bind to something, but SL, you don't have to know what that is. And it's going to start. So let's say the pattern over here is A, G. Okay. So it's going to come it is the DNA polymerase over here. DNA poly. And it's going to start transcribing. So as we go by the law of complementary base pairing, we know A goes with T, C goes with G. So we're going to try to finish this code. T, G, G, T bonds with A bonds with T, C, G, 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 A, T. Okay. Oh, my bad. Okay, oh, my bad. Oh. Oh no, let me just draw a new strand and make our lives easier. Okay, yes. So here is our new strand. It has this code. So these two are complementary. And as you can probably guess, this DNA strand has also has the complementary of this. So we're going to call this the lagging strand, because it's going to take longer to replicate the DNA on it. But this is HL material, so we're not going to talk about that. But you just need to know how the leading strand replicates, and that is how. So DNA polymerase, what did we use here? We used helicase to unravel the tight, helicase and the topoisomerase to unravel the tight hydrogen bonds between the two DNA strands. And then, after we unwrapped the DNA and made it readable, we replicated it by using DNA polymerase, which went and brought some free nucleotides. So these are made up of free nucleotides, which look like this. Okay, yes. So this is a free nucleotide. What's special about them is that they have 3 phosphates. So they're triphosphates. So it'll be something triphosphate. If it's A, it'll be adenosine, or it'll be cytosine, or it'll be thymine, or it'll be guanine triphosphate. And that is what a free nucleotide is. So, free nucleotide. Okay, so yeah. So the DNA polymerase is going to grab these free nucleotides and it's going to bind them in a 5' to 3' direction of the new strand, and in a 3' to 5' direction on the old strand, or yeah, the old strand. Let's leave it like that, old strand. And the DNA polymerase is going to bind to the 3' end of the old strand, and it will start building based on the law of the concept of complementary base pairing, A with T, C with G. And if you're getting confused and you might think that, no, I can't bind A with T because there is no T. Well, no, that's RNA. DNA does have a T, and when we're replicating DNA, we're making new DNA, not RNA. And we're using DNA polymerase, not RNA polymerase. So that is why we use T's and not U's. Yeah. So now we're going to be talking about the polymerase chain reaction, which can be, the acronym PCR. So the aim of this reaction is to amplify small amounts of DNA and make them more. So it's kind of like you put in 1 and you get out 100 with like pieces of DNA. So to do this, I want you to imagine that we have like a little stirring pot in here. Okay, so it's going to be like a kind of a pot, and we're going to be putting some ingredients inside. So what we're going to put is we're going to have some free nucleotides, which we talked about before. And remember, they're little structures that are like this, and they have 3 phosphates. They have nitrogenous base. So these are the ones that we use for building. Over here, remember, we took the free nucleotides and we built them with DNA polymerase. So yeah, those same ones. The building blocks, kind of. We're going to throw some of those in. We're going to throw in a DNA polymerase. But it's not any DNA polymerase. It is a Taq polymerase, which is taken from a little bacteria that lives in really heated waters. And we do this because we use Taq polymerase instead of DNA polymerase, because in the process of PCR, we make the temperatures extremely, extremely hot. So as we know from our enzymes unit, enzymes get denatured in temperatures that hot. That's why we have to use Taq polymerase, which is kind of heat resistant, I would say. So yeah, free nucleotides, DNA polymerase, and we put our DNA and there. And that's kind of what we need for replication. So yeah, we're gonna do that. And step 1 of the PCR process is going to be called denaturation. What we're going to be doing in denaturation is, you know how the DNA strand is like this? In extreme heat, the H bonds that hold these two together are going to be cut. So they're kind of going to end up like this, and they're going to end up as strands instead of connected strands. So single strands instead of double strands. There. That's a better way to put it. So that's the step of denaturation. And we're going to put really heat. We're going to apply heat. And it's going to be extreme heat, which is, reminder, why we use Taq polymerase. Taq DNA polymerase. But something like that. And second step is going to be annealing. So annealing is the action of joining these two strands. So basically we broke the strands and now we're putting the strands back together again. So annealing, we're taking these strands alone and we're adding them together. Alone, together. So we're going to do this by the DNA polymerase or the Taq polymerase. And it's going to come and it's going to make more bonds between them. But to do this, we're going to be using the new nucleotides. So we're going to have more nucleotides, and so we're going to make more DNAs because we have more empty strands and we have other ones to fill them up with. So they're going to have their complementary pairs, and we're going to anneal them to make more DNA using the free nucleotides. Okay, and step 3 is elongation. And this is kind of making more long DNA strands. So elongation is basically elongating. And making the actual strands. And this is when we actually have way more DNA than we started with. So let's say we started with, this is gonna be our new strands. And we know, as we know, DNA is a semi-conservative process, which means that it does conserve a bit and it does have new strands. So elongation, we're going to have way more DNA than we started with initially in the denaturation stage. Because we know that the goal of PCR is to amplify small amounts of DNA and make very many more DNA. So that's about it for this video. Thanks for watching, and I hope you learned something. Let me know if you need anything or have any questions.